Welcome, everyone. I am delighted to bring a panel of talks today to tell you a bit more about conceptual and theoretical issues in the study of and thinking about causation in the context of causal brain circuits. If there's one recurring theme of the symposium, it's really to show a variety of different ways of what it means to take causation more seriously when we're talking about causal brain circuits. So what do I mean by causality here? So if we want to say X causes Y, by taking causation seriously, we mean it in some of the following ways. We mean that changing X must change something in Y. So there's a manipulationist perspective on causation. We mean talking about counterfactual causation. That means, had X been otherwise, would Y still have happened? And it can well be that causes are neither necessary nor sufficient to make something happen. And this is what happens when we deal with, multi-causal systems. And why does this matter? So the reason this problem is important is, for instance, consider a major research endeavor for many of us is in the context of using the research domain criteria or something like this to understand the causes of mental illness, for example. And the topic of how brain circuits are involved in various cognitive, affective, and social processes becomes an important topic where I think it's important to reflect conceptually on what kinds of questions we're asking and the degree to which we need causal answers in asking these sorts of questions. So there's, in recent OHBMs in years past, there's been a lot of progress in neuroimaging on several fronts. We've been improving the kind of statistical modeling we do of functional effective connectivity in brain networks. We've come a long way in how we think about creating encoding and decoding models of behavior and representations of behavior and tackling challenges therein. And there's been some discussion of causality in that context. And we've been doing a much better job of predictive modeling in general, especially in terms of emphasizing generalizability of predictions across samples, tasks, stimuli, populations, and so forth. But I still think there is some more progress to be made from taking our notions of causation more seriously in the ways that I outlined here. And so particularly in this symposium, we're going to ask the following flavor of sorts of questions. And the speakers will address the following flavor of questions in the context of causation. One talk will discuss how brain region X causes brain region activity in brain region Y, for instance. So causation within brain circuits. How can we identify causation within brain circuits? Another question one may ask is, how does brain circuit X cause function Y? And in this context, Paul Cisek will be talking about, what are the different functions and how do we delineate them? For the same kind of questions, another issue that arises is, how are we measuring the behavior or functions Y? And what are the challenges that we face when we don't measure Y appropriately? And another question, more clinically oriented, we could ask is, is brain circuit X a biomarker for Y? And, when do such biomarkers need to be causal? And so our first talk will be Konrad Kording presenting on, causation in the context of functional and effective connectivity. And he's going to argue about taking counterfactual causation more seriously in this context and addressing one of the hidden assumptions that we often ignore in the context of causation, which is ignoring the presence of hidden confounders. Paul Cisek will be telling us about how to carve up behavior from an evolutionary perspective and why the classically delineating functions don't map well onto the brain. And Jackie Sullivan will be telling us about how to probe behavior more effectively, especially in the context of translating findings in clinical neuroscience, translating things from animal models to human models, and what we need to do this more effectively. And finally, I will be speaking about creating a causal taxonomy for the different types of biomarkers that we might find useful for clinical purposes, and when thinking counterfactually is more important for these sorts of questions. And with a little bit of emphasis on what we can do going forward to validate biomarkers from this perspective. So without any further ado, Konrad Kording will be our first speaker. Thank you.
My name is Konrad Kording, and I've been asked to give a talk on what does it mean for brain regions to causally influence one another, and why don't functional/effective connectivity count as causal? Let me first start with the definition of causality. Let A and B be events, causation exists. If we had changed A to A star, the probability of B would have been different. So much of neuroscience is explicitly or implicitly about causality. In the medical domain, we want to know about the causal effect of a treatment. When it comes to more specific treatments, we want to say which changes to a brain would help that brain. It is essential in network science. How does one part of the brain affect another? And it ultimately is the basis of mechanism, which is how parts of the causal chain actually work. So why is this difficult? It would not be difficult if we could produce two copies of the same world, treat one of them, not treat the other, as in the definition, and then see how the future is different for these two worlds. Unfortunately, there's no mechanism in this universe that allows us to do that. The problem really in the real world is confounding. I want to know if one variable affects the other, if the lower variable affects the higher. The problem is there's lots of other variables that affect both variables. And this makes it very hard to estimate if there's causality, if I'm not allowed to perturb the system. So there's a continuum of confounders. There's games like chess or Go or even Atari games where there's no confounders. Then there's domains like medicine where there's countless confounders. Not like how you interact with your doctors, just matters. And then there's brains. Brains arguably have 10 to the 11 confounders. We will almost always record only a tiny part of the brain. So let's get some intuition about what happens because of the dimensions that we don't observe. And let's produce the most simple simulation of a brain that we could possibly think of. Xt plus 1 is matrix A multiplied with xt, and we add some noise, and the noise is Gaussian distributed. And to somewhat model things that look like brains, we make it so that A is a sparse binary matrix, and we scale it so that the largest singular value is 0.99, because otherwise things might explode. Now, it turns out that if we simulate such systems and they're small, think 10, maybe think 30, then correlation and causation is very similar. What that means is that A matrix that we had before is very similar to the time-delayed correlation. And in fact, the R squared in many cases here is 0.8, 0.9. So they're very similar with one another. And here we see why we so often confuse correlation with causation. In many really small systems, correlation and causation are identical. But for really big systems, the two of them become completely different. So why is solving this problem with regression or any other method so complicated? Let's look at regression because it will give us an intuition for that. If we use regression to calculate then we have that our regression coefficients beta are the covariance matrix inverted times the variance matrix inverted times the covariance matrix. Now, what happens if there's variables that we don't measure? In that case, we have yi is xi beta, that's the part that we do measure, plus zi delta, which is the parts that we don't measure, plus ui, which is the various sources, potential drivers. Now we can put that into the equation for estimating beta hat. And what we get is that the expected value there is beta, which is the real causal effect, plus that correction term. This is the famous omitted variable bias equation term. And what we see here is that the estimate of these biases is just of the same. It looks almost the same as the terms that we have for beta, minus the fact that there's this delta in there, which is the correlation, which is the causal effect of the unobserved dimensions. Now, the beta in the calculation has a dimensionality that is proportional, or like there's as many terms in beta as we have observed dimensions. But in the second correction terms, these are all the other unobserved dimensions. And we have one voxel in the brain it might still have millions of dimensions in there. And that means that the second term is arbitrarily bigger than the first, in particular, given that typical neuron correlations are relatively big. So now there's popular solutions that are not really solutions that are used for brain understanding. For example, we can fit and do model comparisons. That doesn't work because the space of potentially meaningful models is just too big. We can use randomized perturbations, but the system is just too high dimensional. However, and there's no way around these problems. But there are possible solutions for certain subproblems. For example, randomized perturbations, if we talk about treatments, are perfectly fine because treatments are usually low dimensional. And then there's quasi-experiments, which is in cases where we have subproblems with known causal structure. So let's briefly review the potential outcomes framework that is popular in econometrics. The idea is for every item that we have, there is a potential future where we get not treated and a potential future if we get treated. And we can then argue about what would have happened otherwise. Let's briefly look at randomized controlled trials. Why do they work? For every item, we'd have what would happen if it gets treated, Y1, and what would happen if it doesn't get treated, Y0. The treatment effect is the average of all those. We can now approximate that as long as we randomly choose which group you're in, and it shows directly then that randomized control trials are correct estimators for causality. Now, there is a continuum of causal validity. There are experimental methods that are called experimental because things get randomized. There's quasi-experiments that I'll get at in a second that are somewhat in between in causal validity. And there's observational studies that only give us very weak hints about how causality may work. And the idea of quasi-experiments is to find something in the world that is as good as random. Here, for example, there's a lot of treatments that have a threshold. For example, if your exam score is above some value, you get a certificate of merit. If it's below some value, you don't get it. We can draw two hypothetical curves. What would happen to every possible student at every possible exam score if everyone got it, and another curve if no one got it. And that discontinuity that we have at 50 at the exam score where you get treated then reveals the real causal effect in the vicinity of the margin. And here's the famous Thistlethwaite and Campbell paper that did that in 1960. On the x-axis, you have the exam score. On the y-axis, you have percent of students later on winning scholarships. And we find these discontinuities, so it actually helps you to have such things on your CV. Importantly, that field has developed methods of doing sanity checking. We can look for cheating, which is visible as a discontinuity of other covariates. Maybe we could, maybe if rich people find a way of cheating, we would find that at the threshold, the number of rich people goes down. And then there's ways of treating fuzziness where compliance is not perfect to these things. So I see potentials for such applications everywhere. When it comes, many treatments have a threshold, arguably the majority. We often do enrollments based on needs. We get grants based on our NIH priority scores being above some threshold. And like just about any recommender system will recommend you certain things based on some internal score. So what's the take-home message? We really mean causality in neuroscience when we ask a lot of our research questions. In many cases, we do not provide any relevant information about causality. Perturbations are the gold standard. Unfortunately, they don't scale. Quasi-experiments are a great set of ideas of how we can approximately get at causality. However, for a lot of the questions that we want to ask in brain science, they cannot be applied. And with that, well, thanks so much for your attention.
Okay, so I'd like to thank Manjari for inviting me to be part of this session. It's just too bad that we couldn't do it in person. So in my talk, I'm going to address the question of what are the brain functions that we seek to find when we look at neural activity. And this usually follows a kind of a classical functional decomposition of behavior into perceptual, cognitive, and motor control processes, which then lead us to define the questions we ask. Things like how do we store and retrieve memories, how do we make decisions, and then to interpret the functional role of given brain regions. And as you know, this is proving to be quite difficult. And I'll describe one example, trying to infer the functional role of the posterior parietal cortex here in the primate brain. This region has for many decades been implicated in attention, but has also for just as long been implicated in intention, the intention to make a movement. Now, this is not just a semantic issue, because these are largely incompatible concepts. Attention is kind of like a filter that brings information, limits the information that enters into cognition for things like memory storage or decision making. Whereas intention is like the output. After you've made a decision, you would tend to perform a function, a behavior. And so it's hard to see how these functions, which have actually been mapped into the same cells, for example, in area LIP, could be done by the same neurons. And the parietal cortex has also been implicated in other sensory functions, in other motor control functions, and in certain types of decisions, again, in area LIP, which raise the questions, what is this area really doing? What is this region really doing? In fact, as you know, many of these concepts don't map so well to the brain. And it's not just a matter of everything being distributed. Very often we have the same circuits doing things that are large, seem conceptually at least very different. So this raises the question, perhaps this is not the right functional decomposition of behavior. And it motivates us to ask, well, where do these concepts come from? In fact, they're very old. The distinction between perception, cognition, and action, I think, actually comes from dualism, a very old philosophical idea that there's a non-physical mind, which forced early philosophers to conceive of interfaces between the physical world and the mind. Perception that presents the world to the mind, and action that plays out the mind's intentions into the world. Now, of course, the concept of the non-physical mind has been largely rejected, and yet the architecture remains. And I think it remains in part because it's simply embedded in the very language that we use for describing behavior. In fact, Russ Poldrack recently estimated that about 80% of psychological terms have already been in use before the year 1800. In other words, long before psychology was actually a science. Again, raising the possibility that this is not the right way to carve up the problem of behavior. So how can we carve up the problem of behavior? Well, the typical approach is a functional decomposition, where we propose distinct information processing functions and then search for specific neural systems that implement them. Perhaps we just don't have the functions defined correctly. What I'm going to describe, though, is a different strategy for doing this, which we can call phylogenetic refinement. The idea here is to actually follow the progressive elaboration of the nervous system over the course of its evolution, letting evolution be our guide for decomposing the problem of behavior. So we ask, what were the stages of neural aberrations along the way, and what were the associated behavioral advances? So to do so, we want to compare across creatures, and then based on knowing their phylogenetic relationships, infer where certain things, certain advances were made. And here's the phylogenetic tree of animals expanded on the lineage that leads to humans. Now, I'm going to go through this very quickly. If you're interested in more detail, much of this is summarized in a paper that came out last summer. So neurons appeared around here, something like the hypothalamus around here, something like a neural tube and spinal cord around here, producing our ancestors, the chordates, which were organized essentially as a tube. Their nervous system was essentially a tube with a frontal hypothalamic-like segment projecting to a spinal cord. And these animals were capable of simple foraging based on projections from the hypothalamus down to the spinal cord. Now, they also possessed an escape circuit involving a structure called the tectum in what would later become the midbrain. This received visual information from a single photosensitive patch in the front of the head and projected downstream ipsilaterally. So that if a shadow fell on the animal, it would quickly scurry away. Now, in early vertebrates, called the cephalids, this single photosensitive patch split into two that migrated to the sides of the head. And the original bilateral projections of the tectum became pruned down to be primarily contralateral. And this was useful because if you have contralateral projections, then if a threat falls on your left eye, you will excite activity on the right side of the body and you'll make it turn to the right. And you'll continue turning to the right until you've oriented yourself away from the threat and then can escape very effectively. Now, this then retinotactile systems expanded, conferring a kind of topography of avoidance actions for specific locations of threats. And part of the tectum, the rostral part, specialized with contralateral projections downstream, which now implemented approach behavior, because now you would turn towards the site of stimulation. And the selection between approach versus avoidance was based on very simple key stimuli, simple rules like if something is really big and it's approaching you quickly, then you want to avoid. Now, amazingly, these circuits have been retained for over 500 million years. So we see them in lamprey, both avoidance and approach circuits. We see them in zebrafish. We see them in mammals, where the tectum is called the superior colliculus. And there is a medial portion that projects ipsilaterally, producing avoidance behavior, mostly from things from above upper visual space, and a lateral superior colliculus that projects contralaterally, producing approach and orientation behaviors. And these two systems face different pragmatic issues. In the avoidance system, you want to have a kind of a mapping between if something's on the left, you want to turn to the right. If something's on the right, you want to turn to the left. If you encounter two threatening stimuli, well, the average of those responses is actually a pretty good response. That actually gets you away from both threats. But that's not going to work in the approach system. In the approach system, if you have a stimulus that's attractive over here, another stimulus that's attractive over here, going down the average response is no good. It's not going to get you anything. In the approach system, you need something else. You need some kind of competition, some kind of winner-take-all dynamics that's going to suppress one of the options in favor of the other. Now, notice you could, at this stage, call this something like attention, because you're going to ignore one stimulus and focus on another. You could call it intention, because you're going to intend to move towards one of these objects. You could even call it decision-making. The point is, of course, these animals, 500 million years ago, didn't care what we would call this today. They just wanted to get at the food. The point is that we can construct a different taxonomy of functions. So we're not looking for distinctions between things like attention, intention, cognition, etc. We're looking for behaviorally relevant distinctions, things like approach versus avoidance circuits, which exist in the superior colliculus still. And this region has been implicated in control of gaze, body orientation, implicated in target selection for eye and head and reaching movements, and many phenomena that one might call attention. Okay, another thing that happened in the early vertebrates is part of the hypothalamus here expanded into what we now call the telencephalon, with an outer segment called the pallium, which received sensory information, and an inner segment, the subpallium, that projected downstream to select different kinds of behaviors like approach versus avoidance. And this system could learn through reinforcement, dopaminergic reinforcement, what kind of things to approach and avoid. And so this was, of course, very important as well. Now this system continued to expand. The pallium specialized relatively early on into a ventrolateral segment that mostly dealt with dealing with the immediate environment local to the animal, and a medial segment that mostly dealt with navigating to new environments. And this medial pallium would ultimately become the hippocampus. Now, jawed vertebrates added a cerebellum. In bony fish, we get a parcellation and expansion of the telencephalon. And when our ancestors got out on land as tetrapods, they encountered a much wider world of demands and opportunities and affordances for action, and expanded their behavioral repertoire, in part by expanding these pallial sectors. Birds and reptiles expanded this portion. Mammals expanded this portion here, the dorsal pallium, which would become what we now call the neocortex. Now, this region is specialized, has specialized subregions for different modalities like vision, audition, etc. And according to Barbara Finlay and colleagues, in all mammals can be subdivided into a dorsomedial spatial system that maintains topography, and a ventrolateral non-spatial system that does not. And visual motor processing in the spatial portion is used a bit like in the tectum for different kinds of action maps, but now to different aspects of the expanding behavioral repertoire, like walking versus biting versus burrowing, etc. And visual projections into this ventrolateral system now are used to identify the kinds of key stimuli that can be used to evaluate what action is best at a given time. Now, to many of you, this might start to sound familiar. This is essentially the two visual systems theory of Goodale and Milner, where there's a dorsal stream that's involved in visual motor control and a ventral stream that's involved in identifying the kinds of cues that help you select between actions and make decisions, etc. This is also the basis of what I and my colleagues have called affordance competition hypothesis, which proposes how the frontoparietal system potential actions compete in the frontoparietal systems through the influence of various selection biases from other regions. Now, what happened in primates is that the neocortex expanded still further. particularly the temporal and frontal lobes, so much so that the whole thing curled around the insula, creating the familiar C shape of the primate brain. But it can still be subdivided into these two sheets, a spatial sheet that has expanded very much now, producing a wide variety of action maps for the wide variety of actions of which primates are capable. And the temporal cortex is expanded as well, going beyond simple key stimulus detection to object classification and recognition and context-dependent processing to make more sophisticated valuation for more sophisticated selection. Now, this is, of course, a very summarized and rapid description of the evolution of the primate brain, but you can already use it to revisit this question that I posed earlier. What's the functional role of the parietal cortex? So the frontoparietal system contains these parallel sensomotor streams for control of different behavior types, as suggested by Mike Graziano, John Kaas, Leah Krubitzer, and others in many species. They observed different streams for things like walking and climbing, reaching, defense, gaze control, etc. Now, if you're a highly visual tree-dwelling animal with a fovea and frontally faced eyes, this part here, this system here, gaze control, takes on a very special kind of executive role because so many of the other behaviors rely on where the eyes are aimed. Control of gaze influences the selection of what to reach, where to go, with whom to interact, etc. And so because of that, this region, which involves area LIP, the parietal cortex, will produce phenomena that will resemble some aspects of attention, but not others, but also will resemble intention, decision-making, sensory motor control, etc. Now, this is described in the paper, if you're interested, paper with some colleagues looking at this. The take-home message here essentially is that we can conceive of a different taxonomy of functions. It doesn't have to be this distinction between things like perception, cognition, attention, etc., which we've inherited from the long history of philosophical thought, but rather looking at different kind of history, the evolution of the nervous system, which we can actually reconstruct to some degree, giving us a taxonomy that respects how the nervous system evolved and elaborating these different control systems. And I would claim that this taxonomy is better because it actually does map to different brain regions in a promising way. So anyway, if you're interested, there's more detail on this paper published last summer, as well as this paper with some colleagues focusing more specifically on the question of attention. And thank you very much for your attention.
Hi, my name is Jackie Sullivan, and the title of my talk is What Kind of Kinds Are Optimal for Causal Discovery and Translational Research? And I'm a philosopher of neuroscience from the University of Western Ontario, and my hope in this talk is to raise some questions for discussion. I apologize, I'm having trouble advancing my slide. Oh, there we go. Sorry. So my plan today is to briefly motivate the talk, and then I'll address two questions. The first question that I'm going to address is what kinds of kinds are optimal for causal discovery and translational research. Then I'll consider what kinds of practices are optimal for causal discovery and translational research, and then I'll conclude. So to briefly motivate the talk, mental illness is a global healthcare problem. According to the World Health Organization, as of 2017, an estimated 792 million people worldwide suffered from a mental or substance use disorder, and this number is expected to rise. And translational research is considered to be key to developing novel therapeutics for treating mental illness. So there's widespread agreement that developing novel therapeutics requires the use of animal models in whom these therapeutics must first be developed and tested. And there are a number of large-scale initiatives over the past decades that have made this kind of claim. So what kind of kinds are optimal for causal discovery in such translational research? Well, there are two options available out there in the literature. The first is the American Psychiatric Association's Diagnostic and Statistical Manual of Mental Disorders, which essentially puts forward a classification system that consists of categories of mental illness that specify symptom complexes for each mental disorder. Examples include schizophrenia, post-traumatic stress disorder, attention deficit hyperactivity disorder, And the aim of the manual and the aim of the categories is what is referred to as inner rater reliability. So just so long as clinicians consistently use these terms when they're working with their patients, then that's considered to be the goal that is supposed to be met by that manual. On the other hand, the Research Domain Criteria Project, which was put forward by the U.S. National Institutes of Mental Health, spearheaded by Tom Insel when he was director, it instead identifies a set of constructs that designate functions that may be impaired in mental illness, and that can be studied across or irrespective of DSM categories. So some examples of functions that are disrupted in mental illness include such things as habit, learning, working memory, arousal, and attention. And so the solution, or RDoc's claim to overcome the limitations, what they regard as limitations of the Diagnostic and Statistical Manual of Mental Disorders, is to put forward these constructs. And the aim is for the constructs to be valid, to actually correspond to kind of real divisions in the causal structure of the world. Now, what are some reasons why RDoc constructs may be superior for translational research? Well, if you think about the Diagnostic and Statistical Manual of Mental Disorders, if you look at individual categories, what you see is there are a number of deficits or impairments in functioning that are included in the symptom complexes. And essentially what RDoc does is extract these kinds of functional impairments from the DSM, and these correspond to the constructs of psychological functions that may be disrupted in mental illness, according to RDoc. So if you take a category like schizophrenia, persons with schizophrenia may suffer from attentional deficits, behavioral inflexibility, memory deficits, and time monitoring deficits. And these may not be consistent across persons diagnosed with schizophrenia. And also, they may be characteristic of other mental illnesses. And so RDoc allows the possibility of a dimensional approach to studying these different functions. Now, the research domain criteria matrix in which these constructs are placed contains three different kinds of things. First, on the left, it contains the constructs, such as acute threat, attention, working memory. And then there are paradigms that are identified as individuating these constructs that are used in experimental contexts to study them and to study the mechanisms by virtue of which these functions come about. And those mechanisms correspond to these different units of analysis that are in the middle of the matrix, from genes to self-reports. Now, what are some potential pros of shifting from the Diagnostic and Statistical Manual of Mental Disorders constructs to the research domain criteria constructs? Well, one thing that's true is that it facilitates translational research. It's far easier to study psychological and behavioral functions in animal models rather than to mimic whole symptom complexes that are characteristic of mental illnesses. And it's easier to detect functions in animal models, or at least we can say easier in scare quotes. It's not undifficult, but it's easier. Also, there's a reduction in causal complexity, right? If we study individual functions rather than whole DS symptom complexes, it seems to simplify the causal structure that we're interested in investigating. And this is a potential response to Konrad's and Manjari's worries that if the scope of causal investigation is reduced, maybe we'll have better luck in individuating causes. However, there are some problems, obviously, with RDoc, and these are only some of them. First, are these the only functions we should care about, the ones that are identified in this matrix? And what is the process for adding new constructs to the matrix? And does RDoc even have the right constructs designating functions identified in the matrix? And who should decide? In response to Paul's worry, does evolution decide on functional divisions? What considerations do and should guide our selection of constructs? Now, what I regard as a fundamental problem of RDoc is that it has an implementation issue. It aims to facilitate translational research, but if you look at the criteria that informed the selection of the the constructs and the experimental paradigms used to investigate them, what you see is that they were consensus-based workshops that essentially focused primarily on human-based tasks without input from animal modelers. One contrast to this is the Centrix initiative, which I can talk about during the discussion, but it did include animal modelers in the discussion about how to study constructs that designate functions that are disrupted in schizophrenia. So I think this consensus-based approach that's essentially top-down is inconsistent with the desire to reduce causal complexity. And it's in fact antithetical to it and to RDoc's translational goals. So this prompts another question. What kinds of practices are optimal for causal discovery and translational research? And I want to put forward an answer. It's cursory, it's streamlined for the purposes of this talk, but it's inspired by translational cognitive neuroscience and also previous initiatives that have sought to facilitate translational research. So translational cognitive neuroscience is an interdisciplinary area of neuroscience that's aimed at identifying the physiological, genetic, and developmental mechanisms that underlie cognitive impairments that accompany mental illness and neurodegenerative disease, and locating effective therapeutics, including pharmacological, behavioral, and environmental interventions. And these include diet and exercise changes. And the aim of translational cognitive neuroscience is to develop testing methods, behavioral assays. These correspond to these experimental paradigms that are fundamental for the Research Domain Criteria Project. And these are used to assess cognition and cognitive impairments and to investigate their mechanisms. And this is first looked at in rodents as a means to facilitate the translation of preclinical findings to the clinic in the form of effective therapeutic interventions. So this is just kind of a schematic drawing, but essentially there are epistemic benchmarks for causal discovery and for facilitating translational research that you find in translational cognitive neuroscience. It isn't just a question of starting with humans or starting with rodents and then moving down or up. It's more a question of actually taking functions that are disrupted in humans, developing tasks for understanding them, and then trying to develop similar kinds of tasks in animal models that can be used in animal models that then would facilitate translation of findings back to the human case. So what's fundamental, what the epistemic benchmarks are in experiments like this, where you have forward translation and back translation of experimental findings, are the following kinds of what I call epistemic benchmarks. I mean, they're epistemic benchmarks that you're trying to hit for these experiments to actually yield translational knowledge. So first, you want construct validity. You want it to be the case that the paradigms or tests that you use to study a given cognitive function in an animal model actually individuates that function, and the same in the human case. Ideally, well, one of the heuristics that investigators in this domain use is face validity. So if the task and the kinds of stimuli and the kinds of responses are similar between the animal modeling case and the human case, this is a heuristic for establishing that the causal processes that are involved are going to be similar across the two contexts. So it facilitates translation. It is meant to facilitate extrapolation from the one context to the other. And the hope is that if you have the same constructs that you're individuating in the two cases, you're actually tapping into the same mechanisms in the brain. But these, and this is referred to as neurocognitive validity, but these aren't the only criteria that are important. You also want it to be the case in your human experiments and your animal experiments that you've eliminated potential confounds that may jeopardize your ability to establish causal relationships. You want to make certain that your animal population is representative of the human population. So you'd like it to be the case that you're including females and males in your animal experiments, for example. You also are interested in issues having to do with predictive validity. Will a treatment that works in the rodent case be beneficial in the human case? And Also important, and this is true of human experiments and rodent experiments, you want them to be reproducible. It's not enough to discover these things in a single lab. You have to have collaboration to be able to discover these things and make these kinds of connections across labs at different levels of analysis. And so this is what I say, and this is what is in my next slide, or represented in my next slide, that achieving these epistemic benchmarks really requires a kind of coordination of practices and perspectives and technical expertise across research groups. And this kind of collaboration needs to be ongoing. You can't just decide that here are the constructs that we're going to look at, and here are the tasks, and not have an interaction between investigators working in different areas of neuroscience that are studying these mechanisms. And so to some extent, it requires a certain amount of grassroots effort on the part of individual investigators working in different labs who share epistemic values in common to work together to facilitate the translational goals that RDoC wants to facilitate. So my conclusions are as follows. The shift from the Diagnostic and Statistical Manual of Mental Disorder categories to the research domain criteria constructs makes good sense from a research perspective, given that developing effective therapeutics for treating functional impairments in mental illness requires translational research. Yet successful causal discovery and translational research requires that certain epistemic benchmarks be realized, and realizing them really requires multidisciplinary collaboration that RDoC on its own does not facilitate. And making progress in translational research can benefit greatly by facilitating conversations among diverse members of the scholarly community to talk about the challenges and to develop collaborative solutions to determine if the sets of epistemic values are shared, and to agree upon what the right strategies are going forward. I've referred to this in my published work as coordinated pluralism, and Manjari brought such a group of us together for this symposium. And I just want to thank Manjari and Narayan for organizing this symposium and inviting me to be a part of it. And I also want to thank members of the Translational Cognitive Neuroscience Laboratory and the Prado Laboratory at Western for influencing my thinking about these issues over the past year and a half. And finally, thank you for listening.
Good afternoon, everyone. So I am looking forward to setting up a discussion on when brain circuit biomarkers need to be more causal. And to do that, I'm going to introduce a more detailed taxonomy of what biomarkers are and how we can apply more causal inferential thinking to thinking about biomarkers and biomarker discovery. So in general, what do we mean by biomarkers in the context of human brain mapping? Well, any imaging-derived biomarker is basically an indicator of a normal biological process involved in explaining psychological behavior or phenotype or a clinical outcome. And typically, what we might do is, if we take the example of fMRI and brain networks, we might take an individual and infer some kind of functional connectivity or effective connectivity in an individual, and then we might want to link variation in networks or features of networks with variation in behavior or clinical outcomes. And any result from this kind of process will give us a biomarker. But the ultimate goal of one of our interests in discovering such biomarkers is to use it to inform diagnosis and treatment of, for instance, psychiatric disorders or neurological disorders. And the current conception of what this looks like is that we might do clinical neuroimaging studies where we do biomarker discovery, as I just mentioned, and then we might proceed to doing clinical trials where we do a series of increasingly difficult validation, including ruling out confounding factors and showing clinical utility and so on and so forth. But an important question that we might ask is, can some of these validation procedures be done at the point of biomarker discovery itself? And that's the perspective from which the rest of my talk will proceed from. And one reason why this kind of take might be important is that a lot of biomarkers that we are going to discover are going to be purely correlative. And we just might not have the sample size to do complete validation of all the possible correlative biomarkers we find in imaging in the context of clinical trials. So for example, a recent study has shown that the sample size requirements for validating biomarkers in a much more stringent and counterfactual manner can be really high. In fact, it's much higher than the sample sizes actually used in a recent state-of-the-art clinical trial in the context of discovering imaging biomarkers. And so I think it's really important to think about what can be done in the process of biomarker discovery that that saves us degrees of freedom and sample size for true clinical validation in the clinical trial phase. To rethink this, I think it's useful to actually dive into what are the different biomarker categories. And this is something we don't do quite enough in the context of imaging. We talk about imaging biomarkers in general, but the questions we ask in the quantitative manner when we formulate our regression or prediction models are often not completely tailored to the biomarker categories. So if you've ever looked at the FDA NIH guide to for defining biomarkers, you'll find that there are several different biomarker use cases, those used for diagnostics, those used for serial monitoring of a disease process or not, those used for predicting treatment response, those used for predicting just risk of developing an illness, and biomarkers used for as a substitute for a clinical endpoint. So all of these are different use cases, and I think it would be valuable to take each of these categories more seriously and to formulate them in corresponding quantitative manners. And to do this, what I will do is apply a hierarchy of the different flavors of causal questions we may ask. So for example, you might have already heard from an earlier talk by Konrad Kording about Pearl's ladder of causation, and particularly distinguishing associational from counterfactual questions. And so here, let me do a quick recap of what the ladder of causation is. So Yuta Pearl tells us that there's actually 3 different flavors of predictive questions one may ask. And the ones we traditionally employ in both statistics and machine learning are purely of the associational flavor. So for example, when we perform a regression or do train predictive models in the context of machine learning, what we're often asking is, what is the probability of a response Y, given that we observe that our predictors X take the value X? An interventional prediction, on the other hand, says, what's the probability of a response Y, given that we set certain predictors to take the value X by a hypothetical intervention, if not an actual intervention. And a counterfactual prediction would in turn ask, what is the probability of a response Y had a covariate or predictor taken the value X, given that what we observe is that the predictors X take the value X prime. So these are the different flavors of predictive questions one may ask, where ultimately there is an association being made between predictors and outcomes, but the questions being asked quantitatively are actually quite different. And in general, there's no reason that the models that we train using an associational prediction type objective will match the ones that come out of a counterfactual prediction objective. And so to hit home why this is important, it's important to realize that in these definitions of associational versus interventional and counterfactual predictions, that the interventions being talked about here are used to define the goal. It's not actually necessary that this means that your study has to involve an experiment. The main differentiation here is that we are changing the nature of our objectives, our goals, by thinking in terms of interventions. And an example from the area of epidemiology for why this kind of distinction matters is that if one considers, for example, trying to understand the effect of hormone replacement therapy in the context of a cohort study rather than a clinical trial, if one formulates the question in an associational manner, you get the wrong answer that hormone replacement therapy actually improves cardiovascular outcomes. Whereas if you were to ask the question in a manner more consistent with counterfactual prediction, then the results will actually match what we get from an RCT, which show that hormone replacement therapy actually makes cardiovascular outcomes worse. So actually changing the way we formulate our questions can actually have an impact on the answers we get. And that's why, you know, changing the objective of how we go about discovering biomarkers can similarly have a beneficial effect. So what would this taxonomy look like if we were to apply it to the context of the biomarker categories I discussed before? So for example, if we consider risk biomarkers, all they need to satisfy is that if somebody has a certain biomarker that increases the likelihood of them developing a disease, then you now have a risk biomarker. And such biomarkers can be discovered by purely formulating the question in an associational prediction manner. So risk biomarkers can be discovered purely by using traditional regression and predictive machine learning when done well, you know, using cross-validation and finding stable generalizable associations. But the question you can ask can be purely associational, and you can still develop them. So for example, hippocampal volume is a risk biomarker for Alzheimer's, and it's one that is successfully used in practice now to enrich clinical trials. And that's an example of a risk biomarker that has come out from imaging studies. On the other hand, if we want to understand or use biomarkers to identify engagement of a therapeutic target, then the question being asked is fundamentally interventional. Similarly, if we ask whether some biomarkers reflects what happens under a clinical treatment setting and so forth, then we're actually asking a question of, you know, what's going to happen in the context of disease progression for an individual under a particular setting if we were to do something. And so several biomarker categories have at least some use cases that are they're asking at least hypothetical interventional questions. Other biomarkers, for example, predictive biomarkers, which are often the biomarkers we are looking for when we say we want biomarkers that identify response to treatment. What we're actually asking is, if an individual were to be given psychotherapy versus a pharmaceutical drug or some other treatment versus a placebo, would this biomarker reveal that this individual would respond better to one particular treatment compared to other treatments or placebo? And this is, in fact, a counterfactual question. So our questions about, for example, finding intermediate mechanisms that can replace biomarkers that can replace directly measuring clinical utility or benefit. And so several biomarkers of the kinds we're interested in are actually conceptually asking counterfactual questions. Yet the procedure that we might use to discover candidates for such biomarkers might actually be currently associational. And the question is, how can we change change our style of analyses for this? So let me take and dive a little more deeply into the case of surrogate endpoints as an example. So a general biomarker, a correlative biomarker, need only reflect the disease. It need not lie on the pathway between disease and true clinical outcome. But for example, a surrogate endpoint, it not only needs to lie as an intermediate, it not only needs to measure an intermediate mechanism between the disease and the true clinical outcome, it also needs to reflect behavior under interventions, and it also needs to satisfy the condition that these interventions actually improve clinical outcomes. So several causal questions need to be satisfied in order for a biomarker to reach the status of a surrogate endpoint. And I think even though clinical neuroimaging studies are not looking for surrogate endpoints, we're often looking for intermediate mechanisms that can be measured by the brain, that come in between the disease and some kind of behavior or symptom phenotype. And so the flavor of questions we're often looking for often resemble that of clinical endpoints, surrogate endpoints. And so at a minimum, even if we're not conducting an interventional study, we should be asking the question, do our biomarkers are they at least causal mediators, or do they at least satisfy the causal relationships that will make them good mediators so that they may prove to be better candidates for clinical validation down the road? An example of where I think this kind of thinking might be beneficial is, for example, finding biomarkers of aging. We're often interested in how aging changes the brain as measured by, for example, fMRI, which in turn measures how we might experience better or worse executive function and other kinds of behavior. And so in this context, the question we're often asking is we're looking for an intermediate mechanism through fMRI, an intermediate mechanism between age and cognition, for example. And so these sorts of biomarkers, I would argue, need to meet the criteria that look a lot more like surrogate endpoints. And so let me just do a brief summary here that this is what that it can be valuable to think about breaking down biomarker categories and thinking more carefully about what our questions are and which ones have a more causal flavor, and try to directly ask those questions in a quantitative manner. And here's an important lesson we might take away from other fields such as cancer oncology, where a lot of candidate biomarkers have been proposed for personalizing treatments in the context of different types of cancers, or defined surrogate endpoints. Nevertheless, most of the biomarkers proposed in this context have always only met associational standards, not interventional or counterfactual standards. And as a consequence, most putative cancer biomarkers ultimately have failed. In fact, work done by Vinay Prasad and others have shown that the number of cancer biomarkers that have shown even any evidence of clinical utility are often much less than 7%. And they directly link this to failure to validate such putative markers using more counterfactual approaches. So what are the challenges to making biomarkers more causal? In the last few minutes, let me briefly summarize an issue that have come up in earlier talks, and I think were also highlighted by Konrad. Konrad highlighted the problem of confounding in the context of functional connectivity, but the same kind of problem arises when we want to link a biomarker X with some kind of symptom or phenotype or clinical outcome Y, which is that how can we be sure that there are not a large number of unmeasured variables U that are common causes of the biomarker and outcome, for example. A second kind of problem that affects or can create bias that I won't be talking about today is actually the problem of collision, or which often takes the form of selection bias. This is where X and Y may not be associated at all, but you're measuring them in some very specific context conditional on some unobserved variable S. And this can create an association when there is none. But I'll just mention that there are other types of bias that are not confounding, but I won't be talking about them for the remainder of the talk. So the question is, how can we be sure that there is no effect of some unmeasured variable U or S, and what can we do to address this? And just to emphasize that this flavor of these two kinds of biases can arise in almost every kind of study design, whether it is clinical trials or epidemiology and so forth, or whatever intervention we might be using. And so there's a flavor of these two problems that can arise in several settings. And so articulating our questions in terms of the causal effects we're interested in is always going to be valuable regardless of whether you're doing a clinical trial or not. But let me just now focus on how we might address the issue of confounding. There's broadly three categories of strategies to address confounding, which is, let's try to find and actually measure the unmeasured variables U and account for it. So a flavor of how we do this with an fMRI, for example, when we do anatomical COMScore correction or physiological noise correction takes this flavor. But then what we never know for sure is whether there is residual confounding, like have we measured all of you, or are we only measuring a subset of you? But there's other approaches we can use to measure unmeasured confounding, which is try to find an instrument Z that directly causes or is highly correlative to the putative cause X, but does not cause any unmeasured, is not at all linked to the confounding at all. And taking advantage of multiple modalities, preclinical studies, genetic information, brain stimulation, they're all examples of study designs that might yield useful instrumental variable Z. There's also a third approach, which in the context of imaging, it's unclear whether these can be found, but the idea is if you're able to find a strong mediator between the biomarker and outcome, then you can take advantage of that to directly account for unmeasured confounding, even if you don't know what it is. But I think these finding such mediators and verifying them tends to be hard in practice. So broadly speaking, there are 3 strategies to account for unmeasured confounding. Sorry. And the thing is, we're only partially exploiting one strategy right now, and we never know for sure if we're doing good enough in one strategy. But if we were to combine multiple strategies, then that gives us an approach for testing whether our assumptions using one strategy are valid or not. Coming back to our example of brain aging, it's possible using multiple modalities or special sequences like multi-echo to actually validate the assumptions regarding whether we are tackling unmeasured confounding in an appropriate manner or whether we need to do more. And this can be quantified by taking advantage of both strategies, whether we find negative controls that directly measure confounding, or we find instrumental variables that that account for unmeasured confounding in a different way. In the interest of time, I'm going to I won't dive further into this example, but you're welcome to ask me during Q&A how this might be achieved in more detail. Summarizing my talk, in general, it would be beneficial for us to think more carefully about different biomarker use cases and take the categories more seriously. And we can potentially do more to eliminate purely correlative biomarkers that are not going to be useful later on in clinical practice by taking advantage of both formulating causal questions directly when we train our models, as well as employing multiple strategies to actually account for the assumptions we might make in trying to solve these sorts of quantitative questions. There's more information about all our talks today at neurostats.org/OHBM2020. And we'll now move into the Q&A session. Thank you.